Analgetik adalah zat yang bisa mengurangi rasa nyeri tanpa mengurangi kesadaran. Nyeri adalah perasaan sensorik dan emosional yang mengganggu, berhubungan dengan ancaman timbulnya gangguan atau kerusakan jaringan. Penggolongan analgetik. 1. Analgetik opiat. Penggunaan analgetik opiat yaitu meredakan atau menghilangkan rasa nyeri, tetapi semua analgetik opiat menimbulkan diksi atau bergantungan. 2. Analgetik non-opiat. Penggunaannya meringankan sebuah trauma, menghilangkan rasa nyeri tanpa berpengaruh sistem saraf pusat, dan menurunkan kesadaran juga tidak menimbulkan ketergantungan. 2. Diberikan untuk nyeri ringan sampai sedang, misalnya nyeri kepala, gigi, otot, atau sendi perut, dan nyeri hati. Selanjutnya, contoh golongan senyawa obat. Analgetik opiat, contohnya morfin, kodein, tramadol, metadon. Analgetik non-opiat ada antiinflamasi non-steroid, antikonvulsif, NMDA antagonis, antidepresan, dan relaksan otot. Kita lihat ini mekanisme kerja dari analgetik opiat. Kemudian kita masuk ke obat. Satu, morfin. Morfin adalah agonis opiat yang memiliki afinitas terbesar pada reseptor mu. Reseptor ini merupakan reseptor opiat analgetik mayor. Indikasinya nyeri berat, baik akut maupun kronik, nyeri yang disebabkan oleh infark miokard, dan nyeri karena kanker. Mekanismenya menghambat sinyal saraf nyeri ke otak sehingga tubuh tidak merasakan sakit. Adapun interaksi obat, jika digunakan bersamaan dengan obat ini, itu obat yang menyebabkan depresi sistem saraf pusat. Obat anti diare, antikolinergi yang digunakan bersamaan dengan morfin. Efek sampingnya ada konstipasi, ketidakseimbangan hormon, dan depresan sistem saraf pusat. Kodein. Kodein merupakan obat pereda nyeri golongan opiat yang dibuat dari ekstrak tumbuhan opium. Kodein meredakan nyeri dengan cara mengurangi respon nyeri yang diterima oleh otak. Mekanismenya bekerja dengan mengaktifkan reseptor mu dan ditandai dengan efek analgesik kerja cepat. Sangat berpengaruh oleh polimerisme CYP2D6 dan terutama dikeluarkan melalui urin. Interaksi obatnya bisa meningkatkan efek samping depresi sistem pernafasan jika digunakan dengan obat-obat anestesi dan antihistamin. Meningkatkan kadar kodein dalam darah jika dikonsumsi bersamaan dengan simetidin. Efek samping yang ditimbulkan yaitu drowsiness dan konstipasi. Selanjutnya ada tramadol. Tramadol hanya digunakan untuk meredakan nyeri ringan sampai sedang dan tidak efektif untuk nyeri kronis. Mekanisme tramadol bekerja dengan dua mekanisme. Pertama, dengan mengikat secara stereospesifik reseptor mu opiat di sistem saraf pusat untuk mengeblok sensasi nyeri dan respon terhadap nyeri. Kedua, menghambat pelepasan neurotransmitter serotonin dan norepinefrin dari sistem saraf aferen yang sensitif terhadap stimulan yang berakibat terhambatnya impuls nyeri. Adapun interaksi obat-obat yang bekerja di sistem saraf pusat seperti transgulinisor, hipotonik, dan sedatif. Karbamazepin mengurangi efek analgetik tramadol, menghambat enzim CYP2D6 dan menghambat enzim CYP3A4. Efek yang ditimbulkan bisa mual, muntah, pusing, mulut kering, vertigo, konstipasi, dan drowsiness. Obat metadon. Metadon merupakan salah satu jenis obat analgesik opiat, yaitu golongan obat pereda nyeri yang menyebabkan ketergantungan jika digunakan secara berulang. Karena itu, penggunaannya harus berada di bawah pengawasan dokter. Mekanisme kerjanya yaitu bekerja pada reseptor opium mu, kapha, dan delta pada sistem saraf pusat dan organ yang memiliki otot halus. Interaksi obat yang ditimbulkan yaitu meningkatkan resiko gejala putus obat, meningkatkan kadar dari resiko efek samping metadon jika digunakan dengan semitidin, meningkatkan kadar diazepam, lorazepam dalam darah. Efek samping yang ditimbulkan perubahan emosi, gangguan penglihatan, gangguan tidur, sakit kepala, sakit mah, pernapasan lambat. Kemudian kita ke obat analgetik non-opiat, tipe antiinflamasi non-steroid, ada aspirin, paracetamol, natrium diklofenak. Indikasinya sakit kepala, sakit gigi, nyeri punggung, pegal-pegal, dan nyeri sendi. Mekanismenya menurunkan aktivitas enzim siklooksiggenase, menurunkan sintesa prostaglandin di jaringan perifer dan sistem saraf pusat. 1. Aspirin digunakan untuk pengobatan demam, meredakan nyeri akut, serta inflamasi. Mekanismenya menghambat secara irreversibel enzim COX. Interaksi obat digunakan bersamaan dengan obat kafein, kadar aspirin meningkat. Tidak boleh digunakan pada anak-anak yang mengalami demam yang disebabkan infeksi virus. Paracetamol digunakan untuk meredakan nyeri ringan sampai sedang, contohnya demam. Mekanisme kerjanya menghambat biosintesa prostaglandin melalui inhibisi koenzim siklooksiggenase di sistem saraf pusat, bukan perifer. Menghambat proses regulasi panas di hipotalamus. Efek sampingnya bisa menimbulkan mual, gatal-gatal, urin berwarna gelap, reaksi alergi. Adapun interaksi obat, jika digunakan bersamaan dengan obat ini menurunkan efek paracetamol jika digunakan bersamaan dengan phenytoin phenobarbital. Natrium diklofenak digunakan untuk mengurangi nyeri dan peradangan. Mekanisme kerjanya menghambat kerja enzim siklooksiggenase. Efek samping yang ditimbulkan bisa sakit perut, gangguan pencernaan, mual muntah, sakit kepala, ruam. Meningkatkan kadar enzim hati dan di darah. Interaksi obat yang ditimbulkan dengan alkohol atau obat golongan antiinflamasi non-steroid lainnya berupa peningkatan resiko efek samping ulkus, peptikum, dan pendarahan saluran cerna. Analgetik tipe antikonvulsif, obatnya ada carbamazepin, gabapentin, pregabalin. Mekanisme kerjanya berhubungan dengan menghambat influx ion kalsium dan pelepasan neurotransmitter eksitasi pada jalur pembentukan nyeri pada spinal dan supraspinal. Indikasinya untuk neuralgia trigeminal dan neuralgia diabetes. Kemudian analgetik tipe NMDA antagonis, obatnya ada ketamin, mematin, dextromethorphan. Mekanisme kerja menghambat reseptor NMDA yang berperan peningkatan intensitas nyeri. Indikasinya nyeri kronik terkait kanker, nyeri pasca operasi. Lanjut ke analgetik tipe antidepresan. Ada amitriptilin dan nortriptilin. Mekanisme kerjanya menghambat proses pengambilan kembali serotonin dan non-epinefrin sehingga meningkatkan kadar keduanya di syaraf tulang belakang. Indikasinya nyeri akut pasca bedah dan nyeri neuropati. Untuk analgetik tipe relaksan otot, contohnya obatnya siklobenzaprin, metaxalon. Mekanisme kerjanya menghambat proses pengambilan kembali non-epinefrin di locus coeruleus yang menyebabkan peningkatan kadar non-epinefrin pasca sinap dan merelaksasi otot rangka. Indikasinya nyeri otot rangka. Berikut contoh obat yang beredar di pasaran yang biasa kita temui. Penyakit Parkinson merupakan suatu penyakit atau sindrom karena gangguan pada ganglia basalis akibat penurunan atau tidak adanya pengiriman dopamin dari substansia nigra ke globus pallidus, menyebabkan gangguan gerakan yang kronik progresif ditandai adanya tremor, bradykinesia, rigiditas, dan ketidakseimbangan postur. Penggolongan obat Parkinson: 1. Obat-obat dopaminergik. 2. Antikolinergi. 3. Obat-obat dopaminergik inhibitory. 4. MAOB inhibitor. 1. Dopaminergik. Meningkatkan kadar DA di otak atau meningkatkan transmisinya, dan dengan demikian berdaya meringankan hipokinesia dan kekakuan, tetapi jarang mengurangi tremor. Contoh obatnya ada levodopa, bromokriptin, dan perangsang sistem saraf pusat. Mekanismenya berdasarkan pengisian kembali kekurangan DA di korpus striatum. Efek samping yang ditimbulkan bisa mual, muntah, nafsu makan berkurang, diskinesia dan gerakan spontan abnormal, dan stres. Interaksi yang ditimbulkan menghambat dekarboksilase yang menyebabkan menghambat biotransformasi levodopa menjadi DA, berguna untuk meningkatkan jumlah levodopa di jaringan otak, dosis efektif lebih cepat tercapai, dan efek samping berkurang. 2. Obat-obat antikolinergi. Contoh obatnya benztropin, trihexylpenidin. Menghambat secara kompetitif reseptor-reseptor muskarinit di dalam otak, termasuk yang terdapat dalam sistem ekstrapyramidal. Efektif untuk mengurangi tremor pada Parkinson. Terapi efeknya tidak kuat pada bradykinesia Parkinson. Untuk Parkinson hanya diberikan per oral. Efek samping yang ditimbulkan sentralnya gangguan neurologi, bisa ataksia, bisa septhia, hipertenia, gangguan mental. 3. Obat dopamin antikolinergik. Contohnya amantadin, meningkatkan aktivitas dopaminergik serta menghambat aktivitas kolinergik di korpus striatum. Efek samping ditimbulkan intoksikasi atropin, disorientasi, gelisah, insomnia, pusing, gangguan saluran cerna, mulut kering, dermatitis, halusinasi, dan ansietas. 4. MAOB inhibitor. Penghambat spesifik pada MAOB bertujuan menghambat proses degradasi dopamin dalam otak dan memperpanjang efeknya. Di samping itu, obat MAOB inhibitor lebih menghasilkan efek samping relatif rendah. Contoh obatnya seleglin. Obat ini digunakan dalam terapi kombinasi dengan levodopa. Seleglin
Mekanisme kerja menghambat deaminasi dopamin sehingga kadar dopamin di ujung saraf dopaminergik meningkat.
Drugs for Parkinson's disease. So let's get right into it. Parkinson's disease is a neurological disorder that results in a progressive loss of coordination and movement. Now, the neurons responsible for coordinating movement are located in a part of the brain called striatum, which receives information from two major sources, the neocortex and a region known as the substantia nigra. The cortex relays sensory information as well as plans for future action, while the substantia nigra sends dopamine that helps to coordinate all of the inputs. Now, Parkinson's disease develops when the neurons connecting the substantia nigra to the striatum progressively degenerate. Since dopaminergic neurons that originate in the substantia nigra normally exert inhibitory effects on GABA neurons located in the striatum, too little dopamine results in more GABA, causing increased inhibition of the thalamus, as well as reduced excitatory input to the motor cortex. In addition to that, these same dopaminergic neurons also exert inhibitory effects on the excitatory cholinergic neurons in the striatum. And again, without sufficient levels of dopamine, the production of acetylcholine is increased, which triggers a chain of abnormal signaling leading to impaired mobility. Ultimately, the imbalance between inhibitory and excitatory activities leads to the manifestation of typical clinical symptoms that include resting tremor, rigidity, postural instability, and slowed movement. The pharmacological therapy for Parkinson's disease is aimed at replenishing dopamine levels, mimicking dopamine's action, or antagonizing the excitatory effects of cholinergic neurons. Now, in order to get a better understanding of the pharmacology of anti-Parkinson agents, first we need to take a closer look at the dopamine-producing neuron. So here, inside this dopaminergic neuron, dopamine is synthesized in a two-step process, starting with the amino acid tyrosine. First, with the help of enzyme tyrosine hydroxylase, tyrosine gets converted to L-DOPA, also known as levodopa. In the second step, the L-DOPA formed by tyrosine hydroxylation is quickly decarboxylated by another enzyme called aromatic L-amino acid decarboxylase to the neurotransmitter dopamine. Dopamine is then loaded into synaptic vesicles and released by physiological stimuli into the extracellular space, where it can bind to dopamine receptors that are expressed on the postsynaptic neuron. Finally, excess dopamine in the synapse is reuptaken back into the neuron or into glial cells, where it gets metabolized by monoamine oxidase, abbreviated MAO, and catechol O-methyltransferase, abbreviated COMT. It's important to note that while the MAO enzyme exists in two forms, known as type A and type B, the type that is predominantly found in the glial cells is the MAO type B. Now let's move on to discussing drugs used in treatment of Parkinson's disease, starting with one of the most commonly used drugs, that is levodopa. But first things first, you may wonder, well, why would you use the precursor of dopamine instead of dopamine itself? And the answer is, it's because of the blood-brain barrier. Blood-brain barrier is a tightly packed layer of endothelial cells that restricts free access of molecules between the blood and the brain. Dopamine happens to be one of those molecules that cannot freely pass through this barrier. However, levodopa can, with a little bit of help. One of the biggest problems levodopa faces on its own is peripheral metabolism. There are two major enzymes in periphery which cause breakdown of levodopa before it can reach the brain. That is peripheral dopa decarboxylase, which converts levodopa to dopamine, and catechol O-methyltransferase, which converts levodopa to 3-O-methyl-dopa. Because of this, levodopa must be administered with another agent, called carbidopa, which inhibits dopamine decarboxylase and thus reduces metabolism of levodopa in the periphery. Another agent that is used in combination with levodopa and carbidopa is entacapone, which inhibits peripheral COMT, and thus, just like carbidopa, it prolongs the time that levodopa is available to the brain. Now, levodopa is carried across blood-brain barrier by amino acid transporter. Once inside the brain, levodopa is efficiently converted to dopamine, thus supplementing depleted dopamine levels in the midbrain. However, let's not forget that dopamine is also susceptible to breakdown by COMT as well as MAOB, which convert dopamine to 3-methoxytyramine and 3,4-dihydroxyphenylacetic acid respectively. So here another useful drugs come into play, namely selegiline and rasegiline, which selectively inhibit MAOB, and tolcapone, which inhibits COMT. As a side note here, in comparison to entacapone, tolcapone can better penetrate the blood-brain barrier and thus can act both in the central nervous system and in the periphery. So again, as you can see, by decreasing the metabolism of dopamine, these drugs help to increase dopamine levels in the brain. Unfortunately, because Parkinson's is a progressive disease, with time the number of dopamine-producing neurons decreases, and fewer cells are capable of making dopamine. Taking that into consideration, some drugs have been developed to mimic dopamine and directly stimulate dopamine receptors in the brain. Drugs that belong to this class include bromocriptine, ropinirole, pramipexole, rotigotine, and apomorphine. Now, as I mentioned at the beginning of this lecture, in Parkinson's disease, dopamine depletion leads to increased acetylcholine release, which then activates muscarinic receptors located on the neurons responsible for smooth motor control. The overstimulation of these neurons by acetylcholine then causes tremors and rigidity. This is where anti-muscarinic agents come into play by blocking the muscarinic acetylcholine receptors and cholinergic nerve activity. As a result, these anticholinergic agents restore the balance between acetylcholine and dopamine, which may improve the symptoms of Parkinson's disease. Drugs that belong to this class include benztropine, bipyridine, procyclidine, and trihexyphenidyl. Now, before we move on, I wanted to briefly mention here one more drug used in the treatment of Parkinson's disease, that is amantadine. Amantadine doesn't exactly fit into any of the classes that we discussed so far. Its mechanism of action is poorly understood. However, some of the speculated ones are that it prevents dopamine reuptake, facilitates presynaptic dopamine release, and blocks glutamate and NMDA receptors. Now, when it comes to side effects, levodopa in combination with carbidopa may cause nausea, loss of appetite, hypotension, mental disturbances, and discoloration of urine, sweat, or saliva. Selegiline and rasegiline may cause nausea, insomnia, dyskinesia, and visual hallucinations. Entacapone and tolcapone may cause discoloration of urine, sweat, or saliva, and diarrhea, which can get severe, particularly with the use of tolcapone. In addition to that, tolcapone has been associated with liver toxicity. All dopamine agonists may cause nausea, orthostatic hypotension, mental disturbances, and daytime sleepiness. In addition to that, bromocriptine has been associated with pulmonary and cardiac fibrosis. Lastly, drugs that block muscarinic receptors generally produce anticholinergic side effects, such as constipation, urinary retention, dry mouth, and blurred vision. And with that, I wanted to thank you for watching. I hope you found this video useful, and as always, stay tuned for more.
Hipnotik sedatif merupakan Golongan depresan sistem saraf pusat, relatif dan selektif, menyebabkan adanya rasa kantuk, tenang, tidur, kehilangan kesadaran, anestesi, dan koma. Indikasinya, sedatif menghambat respon terhadap rangsangan emosi, menenangkan. Sedangkan indikasi hipnotik, dia bisa kantuk, mudah tidur, mempertahankan tidur fisiologis. Adapun indikasi lain, yaitu pelemasan otot, anti-epilepsi, anti-ansietas, induksi anestesi. Klasifikasi 1. Golongan benzodiazepin, contohnya diazepam, klordiazepam. 2. Golongan barbiturat, phenobarbital, tiopental. 3. Golongan lain, meflobaman, klorahidrat. Golongan benzodiazepin, diazepam, klordiazepam. Mekanisme kerja, potensiasi inhibisi neurons dengan GABA sebagai mediator. GABA plus benzodiazepin akan membuka fase dan menyebabkan sel sukar tereksitasi.
In this lecture, we're going to cover the pharmacology of anxiolytic and hypnotic drugs. So let's get right into it. The primary use of anxiolytic and hypnotic drugs is to treat anxiety and insomnia. Anxiety is generally defined as an emotional state characterized by excessive feelings of worry, fear, and nervousness, while insomnia is generally defined as the inability to initiate, maintain, or achieve restful sleep. Now, in order to gain better understanding of how anxiolytic and hypnotic drugs work, first we need to discuss the role of GABA in the central nervous system. So, gamma-aminobutyric acid, or GABA for short, is a major inhibitory neurotransmitter. What that means is that when GABA reaches and binds to a specific receptor located on a neuron, it reduces the activity level of that neuron and makes it less likely to fire its action potential. Now, GABA is recognized by both ligand-gated and G-protein-linked GABA receptors. In this lecture, we are going to focus on the ligand-gated GABA receptor called GABA-A, as this is the main target of many anxiolytic and hypnotic agents. Now, the GABA-A receptor is made up of 5 subunit proteins arranged to form a central pore. These subunits have been labeled with Greek letters such as alpha, beta, gamma, and delta. The subunit composition of GABA-A receptors in the brain most frequently consists of 2 alpha subunits, 2 beta subunits, and 1 gamma or delta subunit. Furthermore, for each subunit, many other subtypes exist. The function of each GABA-A receptor depends on which subunit it contains. For example, GABA-A receptors containing alpha-1 subunit appear to be involved in sleep, while those that contain alpha-2 or alpha-3 appear to be involved in anxiety. Now, when 2 GABA molecules bind to the GABA-A receptor, conformational change is induced, which results in opening of the central pore that allows negatively charged chloride ions to rush into the cell. This in turn increases the negative charge inside the postsynaptic cell, which makes more difficult for an excitatory inputs to cause an action potential. Now, GABA-A receptors are present in high concentration in the limbic system. One part of the limbic system is the amygdala, which contains many neurons that use the neurotransmitter GABA to control feelings of fear and anxiety. In times of stress, excitatory neurons in the amygdala fire rapidly, sending excitatory signals to other areas of the brain. It is this type of firing that leads to feeling of panic or fear. The inhibitory interneurons in the amygdala regulate these emotions by releasing GABA. The release of GABA and its binding to postsynaptic receptors such as GABA-A inhibits the excitatory signals that result in feelings of anxiety and fear. GABA thus has a calming, tranquilizing effect on our emotions, and prevents us from becoming overwhelmed in stressful situations. Now, in addition to a distinct binding site for the neurotransmitter GABA, the GABA-A receptor has also a site at which anxiolytic and hypnotic drugs bind. These drugs can be divided into three groups, that is, benzodiazepines, barbiturates, and non-benzodiazepine hypnotics. Let's now discuss these one by one in detail, starting with benzodiazepines. So, benzodiazepines get their name from their chemical structure, which includes a benzene ring fused to a diazepine ring. Their binding site appears to be the region between the alpha and gamma subunit on the GABA-A receptor. It's important to remember that benzodiazepines do not bind to the GABA recognition site located at the junction of the alpha and beta subunits. Thereby, they don't induce opening of the channel as directly as GABA. Instead, the binding of a benzodiazepine to its receptor site increases the affinity of GABA for the GABA binding site. This leads to increased frequency of the channel opening and in turn increased influx of chloride ions at the postsynaptic cell, which ultimately makes difficult for any excitatory inputs to cause an action potential. So when the excitatory signals caused by fear or stress are inhibited in this way, the result is decreased anxiety and greater feeling of calm. In addition to that, benzodiazepines can also produce mild muscle relaxation as a result of their action on GABA receptors in the spinal cord, cerebellum, and brainstem. The examples of benzodiazepines are alprazolam, chlordiazepoxide, clonazepam, diazepam, lorazepam, and temazepam. When it comes to side effects of benzodiazepines, the most common ones are related to their sedating and muscle relaxing action. They include drowsiness, dizziness, decreased alertness and concentration, as well as impairment of motor coordination. Now let's move on to the next group, that is barbiturates. So barbiturates bind to GABA-A receptor at the alpha or beta subunit, which is different from the GABA binding site and also different from the benzodiazepine binding site. Now, just like benzodiazepines, barbiturates enhance the effect of GABA at the GABA-A receptor. However, instead of increasing the ion channel opening frequency, they increase the ion channel opening duration. In other words, unlike benzodiazepines, barbiturates hold the ion channel open continuously for long periods of time. In addition to that, barbiturates can decrease the activity of excitatory neurotransmitters such as acetylcholine and glutamate, and at higher doses, they can also mimic GABA and directly activate GABA receptors, thus leading to profound central nervous system depression. This is why barbiturates have a much lower therapeutic index than benzodiazepines, meaning the toxic dose is not much higher than the therapeutic dose. The examples of barbiturates include pentobarbital, phenobarbital, and secobarbital. When it comes to side effects, barbiturates most often cause dizziness, lightheadedness, sedation, as well as memory and attention impairments. Furthermore, because of their narrow therapeutic index, it's important to remember that even small barbiturate overdose can result in coma or death due to respiratory depression. Now let's move on to our last group of drugs, that is non-benzodiazepine hypnotics. As I mentioned at the beginning of this lecture, GABA-A receptors in different areas of the central nervous system consist of various combinations of the subunits. The specific subunit that appears to mediate sedation is the alpha-1 isoform. So as their name suggests, non-benzodiazepine hypnotics are structurally different from the benzodiazepines, and as you may already guess, they selectively bind to the alpha-1 subunit of the GABA-A receptor, thus triggering chloride channel opening with resulting membrane hyperpolarization. Because alpha-1 containing GABA-A receptors are highly expressed in wake-promoting regions of the brain, the preferential binding of non-benzodiazepine hypnotics to these receptors results in sleepiness but not anti-anxiety effect. The examples of non-benzodiazepine hypnotics are zolpidem, zaleplon, and azopiclone. When it comes to side effects, non-benzodiazepine hypnotics can cause cognitive impairments, including memory loss, daytime sedation, as well as impairment of motor function. And with that, I wanted to thank you for watching. I hope you enjoyed this video, and as always, stay tuned for more.